Hey guys! Okay, this video is all about pleiotropy, epistasis, sex-linked, and polygenic traits. And those are really confusing terms because they seem like they look the same, polygeny, pleiotropy, but they mean widely different things. So let's work through those one by one because you need to know them. So pleiotropy, the example that's given to you in the slides, it's about, it's true for all pleiotropic effects. One gene has many phenotypic effects, right? Multiple effects on the genotype, on the phenotype, sorry. And in the case of the slides, they show you the Marfan syndrome, when several biological pathways are affected by one gene. And if you have a mutation, you have a lot of complications coming up. I want to talk to you about a different type of pleiotropy. So if we talk about the pleiotropy over time, one of the big reasons why we age is pleiotropic effects. So let's talk about it. Birth to death, that's life. Boom, done, life answers. But seriously though, birth, you have plenty of genes that are active during our early ages, right? Maturation, all of those genes, and even like our drive to have a lot of babies, a drive to meet other people, social traits, and all of those, they induce a lot of positive effect early in life because you get a lot of improvement from that. You get to pass genes to the next generation, and that's what life is all about. So you have a lot of positive effects early on in life on your phenotype and on your fitness, on your outcome of life in terms of genetic outcome. So those beneficial effects, they take a lot of energy, right? You go through a lot of mitosis, you go through a lot of meiosis, you go through a lot, you put your body through a lot of strains, right? So you can think of one of those as human males having a lot of muscles and musculature and being able to use it and fight. Well, it's beneficial early in life because you can survive. Later in life, you fought a lot, you got wounded, you got a lot of damages. Later in life, you're probably not going to live that long. You're probably going to have complications from those wounds. If you have a knee that's hurting, well, your knee's going to hurt more and more when you're older. And that has really detrimental effects and may bring death closer. And that's one of the pleiotropic effect of those genes. Genes that give you a lot of muscles early on, allow you to do a lot of stuff, but then later in life has some cost. And it's not just that, it's also reproduction. Female going through a lot of reproduction, making babies is extremely costly on the body, has a lot of benefits when you make them, and later in life, it has a lot of cost. And what I'm telling you here is that pleiotropy is one of the reasons why we age, because we invest in things early in life that are selected for, because it's beneficial. But once you reproduced, once you've done your job and the next generation is done, all those genes may have detrimental effects later on, but those are not selected against. You already did your job. So all of those accumulates, and by the end of your life, you have a lot of detrimental effects piling up. So that's one other type of pleiotropy, to get you thinking about a different pleiotropy over time rather than in one individual with different biochemical pathway. I just thought I would give you a different example. Switching gears now. Okay, so epistasis. The example that's given to you in the slides, it's all about the blood type, right? And the markers that are on the surface, so the antigenes, the markers on the surface of the blood cell, A, B, or no markers. If you have A or B, they need to be attached and connected to the blood cell. That connector is coded by another gene. If that gene doesn't express itself, you cannot attach that surface molecule. Therefore, you turn out to be a blood O, type O blood. And that's another reason you can have a type O, although you have the genes for A or B, or A and B. Okay, the alleles for that. Okay, another type of epistasis that has shook off the world of DNA. This is a zoomed in on the nucleus. You have your DNA transcription into RNA. The RNA now is trimmed, all the exons and all introns. Then now the coding part is transferred out. You have a ribosome and then protein production. Great. Normal transcription, translation. Now, did you know that a lot of DNA sequence produces RNA that then will go back and guide the expression of other genes. We literally go back to the DNA and guide DNA polymerase to produce, and RNA polymerase guide the expression of genes to make more RNA for proteins. So some genes that may be here on the DNA, their expression controls the expression of other genes up there. So not only 98% of our genome doesn't code for proteins, but although there's 98%, some of it codes for epistasis, for effects of some genes on other parts of the DNA. And when we realize that additional layer on the DNA, that you don't have simple transcription, translation, sometimes you have transcription, going back for a new transcription, enhancing transcription, suppressing transcription of other genes, that blew the mind of scientists because they realized there's more to it than just directly producing protein. There's a lot of regulation that takes place, and all of that is epistasis. One gene expression influences another gene expression, and that's that loop here, that epistasis loop. And I just wanted to bring it to you because I think it's a super cool discovery from recent biology research. And that's one of the three really cool things. The first was transcription translation with the shape of DNA. The second is epistasis. And the third one is realizing that that DNA molecule has a different orientation and different shapes that changes the expression of genes. It's another time of mind-blowing facts. But epistasis is not just physical expression, like the attachment of a blood type molecule from the surface molecule. It can also be just gene expression. Okay, let's switch to sex-linked. So to understand sex-linked traits, it's important to realize what is that homologous pair of sexual chromosome. You have the X chromosomes, that is huge, big, with branches. That's a replicated X chromosomes, right? And you have if you're female, you have two of those, two X's, and if you're male, you have one X, and then you have a Y that is a little chromosome down there. So, things to realize, what we think is that the Y chromosome is simply an X chromosome that has shedded a lot of information. So now you may be like, a lot of people have like not funny jokes at all, like, oh, male can go without all that information, so male are better, like all that crap. That is not what we're talking about. That shedding occurred when it was a long time ago. We're talking about when sexual reproduction evolved. So we're talking about when you needed to have two sexes. It's talking about cells. There's no individual organisms at that point. Okay, so with that out of the way, we can focus on what does that mean. Well, I can give you a hint. I'm not saying it's the only explanation. I'm just giving you something to conceptualize it. So you may, if you start digging into those subjects, you will find that what I'm saying now may be a short-hand explanation. But let's tell it to you. Males produce testosterone, right? Females do too. Females just transform that testosterone in estrogen. Those two hormones, they come from testosterone. So the missing information here that is present for female is probably what allows female to transform that testosterone into estrogen. Males don't have it, therefore don't express it, therefore get stuck with testosterone and its effects, right? So that's a little bit of what's happening with sex links. So when you're missing pieces of it, you don't express them. And of course, that Y chromosome has less space. So all the genes that are situated, and those are replicated chromosomes, all those genes that are situated here, females have two copies of it, because those two copies are the same, they replicate chromosome, right? So potentially the female can have two different alleles, allowing a healthy carrier to be around, dominant gene and recessive disease gene, but the male has only one copy. So if it carries the one copy, like that's replicated once again, so it's the same. If it's carrying the allele that's for the disease, it's expressing it. And that's really the crux of it. So when you draw Punnett squares with X and Y, usually you use the notation of X and then whatever allele you have here, for example. And if you're a male, you would just do X and then you just have Y and nothing in there. Usually people put a dash. But that's it. That's it for sexual link. I just thought I would talk a little bit about it from that perspective. Okay, this video is going to be running a little bit longer, the past 10 minutes, but hang on with me. This is really important. Polygenic traits. I want you to pay attention here because that's a really important part that will be at the exam. So let's talk about it. We talk about this, the skin coloration example in the slide, so I'm going to leave that explanation over there, and I want to talk here more about another example, which would be height. So if you think of all, and I'm going to just squeeze your brain a little bit by changing things around and having the y-axis on the right, because there is no reason why it's on the left all the time. It's just me being contrarian. So if you have here the height of people, and here you have the frequency. Sorry, let's put the height here because that will make it. I don't want to go into that mind too far into changing things around. Let's just put the y-axis on the right for once, and that's all. So the frequency of people in the population having a certain height, and here the different heights coming from really small here to really big here, people. So what you see in a normal population, lots of people have average heights. And if you break it down in different categories, you will see something like that. Lots of people have average heights, and then a small amount of people have really small heights, right? And a small number of people have really high heights, thing like that. So that's the amount of people in the population, frequency, and small height, big height. Most of the people fall in the middle, right? And each of those bars here, it's different heights. So let's see, that's like 5'7", and that's 5'6", right? All the people between 5'6", 5'7", that's that bar, and this is like, I don't know, 7'1", and 7'2". And the people between 7'1" and 7'2", that's that bar, right? And if you fill in the entire graph, you'll see something, and I'm not going to make all the bars, but you see something that's filling in. And usually, although it's not correct mathematically, usually people just use that normal distribution slope to show it. Although you need to know that behind it, you have to count individuals, so there's not a line, that's a bunch of bars. Okay, now that we know that, when you see the distribution of height in the human population, it's crying out loud, screaming at your face, telling you several genes are controlling height. Because if it was one gene, you maybe have like 3 or 4 alleles, so you will have 3 or 4 bars. Maybe 8 if you have a combination of alleles or the intermediate phenotypes, you know, with incomplete dominance between alleles. But you will end up with a number of bars that would be really discrete, not that many bars. You will have people of 5 feet, people of 6, people of 7, and some people in between. That's it. Whereas it's the distribution of height. You can have individuals with any type of height, quarter inches, and everybody is different. That is telling that several genes are controlling for that, similar to the genes controlling for skin color. So that means we all sharing the same genes. We just have different alleles for it. That's exactly the same as skin color. Polygenic traits are affected by several genes, not just one. Not just 2, like 6, 7, 8 genes. Height is a really great example of it. So literally, if you see a ton of bars, a lot of discrete categories of people along a trait, it tells you that that trait has several genes controlling it. Because if the trait is you either have green peas, green seeds, or yellow seeds, that's one gene. 2 different alleles. One gene can have 6 alleles, like the eye colors, and then you end up with, I don't know, 6 or 7 different types of eye colors. But you don't end up with like a ton of eye colors. Although eye colors, you can argue there's a lot of different composition of eye colors and different hues and stuff. So there will be also probably several genes controlling that. But that's the key you want to think about. Several categories, lots of categories of different types of a trait. It's polygenic, several genes are controlling that. If you see 1 or 2 bars or 4, it's probably monogenic trait. Okay, and that's it. There you go. See you next time.